Howdy, Bio 150 folks. So we're going to pick up where we left off. The last few lectures have been talking about water balance and osmoregulation, and now we're going to switch gears a little bit and talk about how organisms control their temperature, or thermoregulation. Remember that thermoregulation is just the ability of an organism to keep its temperature within a certain sort of range that is ideal for that organism to survive at. So just like humans have to keep their temperature at 98.6 degrees, many other organisms also have an ideal temperature that they work at. And temperature matters for a lot of essential life processes. So this is a very sort of simplified diagram looking at how temperature can affect a lot of different important reactions, whether we're talking about growth rate, the ability of enzymes to work in chemical reactions that are important for your survival, whether you're talking about rates of respiration, so cellular respiration, or if you're a photosynthetic organism, rates of photosynthesis. All of these things can be affected by the temperature and have sort of ideal temperatures that they can operate at. So once again, this is a very simplified diagram, and this doesn't represent all organisms. This is probably just one or a few types of organisms. Where these things sort of peak can range for a lot of different organisms, but you get the idea here that temperature has an effect on a lot of different important processes. And environmental temperature affects all organisms, such that some species really specialize or can be specialized for a particular temperature. So whether you're talking about cave-dwelling salamanders that live at 55 degrees most of the time because caves have a very stable temperature, or maybe prokaryotes that live in deep waters off the Arctic Ocean that stay at temperatures of around 4 degrees Celsius year-round. So no matter what you're talking about, some organisms can specialize for a very particular temperature, whereas other organisms, like you might think about a tree in a seasonal habitat, can tolerate a broad range of temperatures. Okay, so for example, this cherry tree might be able to live in really cold temperatures or survive in really cold temperatures, and also in the warmer temperatures of fall and spring, or the really hot temperatures of summer. Whereas these specialized organisms, although they can live at maybe a really cold temperature or 55 degrees year round, and they're specialized for those temperatures, and they can't survive or tolerate being outside of those ranges. And for most organisms, their internal temperature is going to match their environmental temperature. So most organisms are kind of like this lizard that you see here in this thermal image. So this is a human's hands, this is a lizard sitting in that human's hands, and you can see that this lizard pretty much matches the background temperature of the external environment. Maybe it's a little bit lighter and a little bit warmer because it's picking up some heat from that human's hands. But for the most part, most organisms that we know of on Earth are like this lizard in that they match the external environment. However, there are some exceptions, like humans, other mammals, and birds, and a few other species that can actually maintain a higher stable internal temperature no matter what the external temperature is at. Like this lion, for example, that's a mammal, and you can see from this thermal imaging, it's much warmer than the background environment temperatures are. And we'll get into the details of how that works in a little bit. So when we talk about temperature mattering, I want to talk about why it matters initially at the cellular level. So this won't cover everything that's affected at the cellular level by temperature, but two important things that I want to talk about that are affected at the cellular level. And that is plasma membrane permeability and protein stability. So remember, as things heat up, that thermal energy can be added onto or go into and sort of speed up movements of molecules and actually break weak bonds in molecules. So when I say weak bonds, I'm typically talking about non-covalent bonds. So these are things like hydrogen bonding and things like that. If you think back to your chemistry classes, these are weaker bonds. And when you heat things up, basically those weaker bonds can be broken. And so if you heat things up like a plasma membrane, it can split apart like this when it gets real heated up. And also you can have proteins denature. And so heat can break bonds apart, and cold can allow actually stronger bonds, or the non-covalent bonds, to be stronger and allow more of those bonds to form, creating more rigid structures. So let's talk about what I mean in a little bit more detail. So temperature affects membrane permeability. So we're talking about plasma membranes. Remember that plasma membranes are sort of like the security guards of the cell that allow what things can move in and out of the cell by moving through the plasma membrane. And so plasma membrane permeability is very important for cells to move things in and out of them. And membranes are affected, or their permeability is affected by temperature, such that as you increase temperature, you increase membrane permeability. So as you get to a higher in temperature, they're more and more permeable. So they have a little bit less security going on. Things can move in and out of the cells more easily or through the membranes more easily. Whereas at lower temperatures, basically these bonds get much more strong in a plasma membrane. They stick together more quickly. The membranes themselves become more rigid, and it makes the security much tighter for a cell, in the sense that because the membranes are more rigid, it's harder and harder for things to move in and out of the cell or through the cell membranes. And if you get down to 0 degrees where organism membranes freeze, typically an organism dies. So there are some exceptions to this that we'll talk about later, but once a membrane freezes, you're done. And in addition, if you get to really hot temperatures, membranes and their bonds begin to completely break down, and the organism typically ends up dying if the membrane completely disassociates. And there are some exceptions to that too that we'll talk about. So when we talk about extreme heat, so here we're talking about this sort of circumstance right here. Extreme heat can basically separate the lipid bilayer of a plasma membrane. So without extreme heat, this is a plasma membrane, or this is a cell membrane. And if we look at a cell membrane, it's basically made up of these fatty acids that have these hydrophilic heads. And so these are the outside, this is the outside of the cell, this is the inside of the cell, and all these little hydrophobic tails interact with each other, and they have little hydrogen bonds, basically. They're weak bonds that sort of have them stick together or help them to stick together. But if you heat a cell membrane up a lot, what happens is these weak bonds break, and the cell membrane, it ends up sort of stripping or peeling apart, and it becomes essentially non-functional, and the organism will end up dying as a result of this. Now, there is the exception with this little asterisk here that some thermophilic organisms, so organisms that can live in really hot temperatures, have adaptations to prevent this from happening. For the most part, though, this is what happens with extreme heat, is those bonds break, the membrane peels apart, the organism dies. Okay, so what happens to cell membranes at extreme temperatures? So this is just a review of that previous slide. How would you expect heat to affect the permeability of cell membranes? And what about at really cold temperatures? So how would you expect heat and cold to affect cell membranes? So you might want to hit pause right here if you want to take the time to answer this. Otherwise, I'm just going to go ahead and show you the answer. So the correct answer is membranes are more permeable at high temperatures and less permeable at low temperatures. So this is just temperature, this is membrane permeability. And as you get hotter and hotter, basically the molecules move faster and faster, and the membranes start to break down. So you can think of a membrane being sort of like a wax candle. If you keep a candle at a cold temperature, it's really hard and stiff. If you heat it up a lot, it starts to melt and break down. And that's the same way that membranes behave. So beyond just the individual membranes themselves, the composition of the membrane or the lipids that make up a membrane also affect permeability. And so you can have unsaturated fatty acids or saturated fatty acids that make up a membrane. And when you have these unsaturated fatty acid tails on the sort of the molecules that make up a cell membrane, you can see that you get all these little kinks in here. And what those kinks do is they create more space. And basically, it's easier for things to move through a cell membrane that's made up of unsaturated fatty acids, as opposed to saturated fatty acids have these very straight shape. There's no kinks, and so they align up much more closely. Because of that, they link together much more strongly, and they're less permeable. So just to reiterate, a lipid bilayer with no unsaturated fatty acids, so all of these are straight tails, there's no kinks in them, they're much more tightly packed, overall has less permeability, as compared to a lipid bilayer that's made up of unsaturated fatty acids. There's lots of these kinky tails, things aren't packed together as tightly, and so overall this membrane has higher permeability. So given that, basically, the proportion of saturated versus unsaturated fatty acids have been selected to differ in warm-loving versus cold-loving organisms. So, for example, an equatorial prokaryote, so a prokaryotic organism that has a cell membrane, is in a warmer climate at the equator. What type of fatty acids would you predict to have in their plasma membrane? Let's go ahead and hit pause if you want to think about that. Otherwise, I'm going to say the answer now. You would have more saturated fatty acids, okay? So why saturated fatty acids? So if you're in a hotter environment, basically this lipid membrane, because it's in a hotter environment, the heat is going to make the membrane in general more permeable. And so the tails want to counteract that increase in permeability from being in a hot environment by having more saturated, with more saturated fatty acids to make sure the permeability stays lower overall in that hot environment. So let's try this one again. Go ahead and hit pause to read this question and answer it. Okay, so cold, as we said before, tends to make cell membranes less permeable or more rigid. So if it's a cold temperature, the membrane permeability is going to go way down. So you're down here somewhere. And so cell membranes of organisms live in colder climates should tend to have more unsaturated fatty acids. So here we're looking at this scenario. With unsaturated fatty acids, there's more kinks in the tails. Overall, the membrane has higher permeability. So even though we're in a colder temperature that's reducing overall permeability, those kinky tails are counteracting that by allowing the membrane itself to have an overall higher rate of permeability. And so it sort of evens itself out by having these unsaturated fatty acids in that cold temperature. Lastly, the length of hydrocarbon tails also affects permeability, such that longer tails are less permeable. So for example, if you look at the fatty acids that make up butter versus the fatty acids that make up beeswax, those fatty acid tails that we're looking at here, that is these sort of little zigzags, they're much longer on beeswax than they are on butter. And because butter has these shorter saturated tails, those shorter tails make the butter softer, and overall it's more permeable. Whereas beeswax, it has these longer tails that makes it harder and stiffer and overall less permeable. Okay, and so the same thing applies if you're talking about a plasma membrane. So if you have longer tails on a plasma membrane, there's going to be less permeability overall. So given that, which of the following characteristics make a cell membrane more permeable? So go ahead and hit pause now if you want to answer this. Otherwise, I'm just going to go ahead and answer it. So, saturated tails make things less permeable. So if you got the saturated situation, this is what you're looking at. So overall, it reduces permeability because it's more tightly packed. Unsaturated hydrocarbon tails is this scenario right here. So here you're going to be less closely packed together. It's going to be more permeable. Longer hydrocarbon tails, it's going to be less permeable because it's basically a greater distance that if something has to travel to go through the plasma membrane. And on top of that, there's going to be more hydrogen bonds in between here just because it's a longer tail. Whereas if you have a shorter hydrocarbon tail, the opposite is true. It's going to be more permeable. So B and D are going to make a cell membrane overall more permeable. So just as a quick summary, for cold adaptations, cell membranes tend to be shorter and unsaturated to make basically the cell membrane more permeable in this colder habitat, whereas cell membranes in warm habitats, like maybe this hot pool that you're looking at right here, you're going to have longer and more saturated fatty acids in the cell membrane to make it less permeable because it's in that hotter environment. So the next thing I want to talk about real quickly is proteins. So recall from Bio 140 protein structure. So remember, proteins are a linear series initially. The primary structure is a linear series of amino acids. Those amino acids then form this secondary structure, and that secondary structure is formed based on weak hydrogen bonds that form alpha helices and beta pleated sheets. And then lastly, you get this 3D tertiary structure where these alpha helices and sheets basically fold up even more to make the unique protein structure. And this unique protein structure is extremely important for how that protein functions. So the structure dictates the function of the protein. And so basically, this 3D structure that you're looking at that dictates what the protein does and how the protein functions is held together mostly by non-covalent weak bonds. And so what happens is if you go into or put that protein into much higher temperatures, that protein can do what's called denaturation, or will denature. And when it denatures, basically what you're doing is those weak bonds that are holding this normal protein in its functional 3D structure, those weak bonds break. As a result of that, the protein denatures or sort of unfolds, And once it unfolds, it no longer has that important 3D structure that it needs for it to function. And so this protein is no longer functional. This denatured protein is no longer functional. And so higher temperatures are really bad for the function of proteins, typically. And some organisms have evolved cellular level adaptations to deal with these higher temperatures. Specifically, they've evolved to put higher or stronger bonds like ionic bonds or covalent bonds in the sort of secondary and tertiary structure of the protein, so that when that protein gets heated up, so for example, you have this disulfide bond and this ionic bond, these are weaker than those stronger than those non-covalent bonds. That stops this denaturation process from happening when the protein heats up. And we've used this for our own uses. So for example, in thermal hot springs, we've been able to isolate this bacteria called T. aquaticus. And T. aquaticus has this heat-resistant polymerase called Taq polymerase. So Taq polymerase hopefully sounds familiar to you. Taq polymerase is the PCR reagent that we've used in lab to do PCR. Basically, this is the DNA polymerase that we use during the denaturation step and the elongation step in PCR, where basically because this Taq polymerase has a lot of these sort of stronger bonds, it doesn't denature in really high temperatures. And so because of that, we can use this Taq polymerase at high temperatures to do PCR to make a new sister strand of DNA at these really high temperatures where other polymerases would typically break down and denature. The Taq polymerase doesn't. And because of that, we can basically take this Taq polymerase that we grow and isolate from T. aquaticus grown in the lab. We can take that Taq polymerase, put it into our tubes for our PCR reactions, and use that to copy DNA at high temperatures when that DNA is denatured, and typically all the other polymerases from normal organisms that didn't evolve to grow in these hot pools, those polymerases would normally break down. So summary of cellular level adaptations. In addition to the cell membrane adaptations we talked about, in addition, some organisms have more stronger bonds in their proteins to reduce this denaturation.